Hello students, today I am talking about the receptor concept, final part of it. In the previous parts of pharmacodynamics, you have understood the background of the receptor concept, and in the last video you have understood the receptors, cell communication, and you have seen the GPCR structures. Here I'll begin my talk with the various pathways or mechanisms what the G protein coupled receptors are following. So, these are the three different mechanisms by which the thousands of drugs which are following the GPCR pathway will follow any one of this. If you know all these three mechanisms, you will be able to explain the mechanisms of action of those various drugs. So here the first one is the adenylyl cyclase and CAMP pathway. Here adenylyl cyclase is the effector enzyme and CAMP is the second messenger. In the second phospholipase C IP3-DAG pathway, here phospholipase C is the effector enzyme and inositol triphosphate and diacyl glycerol both are the second messengers here. Third pathway what some of the GPCR receptors are following, that is channel regulation, and in this pathway there is no need of second messenger. They directly have the control over channel opening. So let's see one by one these mechanisms and understand how this all happens. First one is adenylyl cyclase CAMP pathway. Now this diagram in front of you, this is I have taken from the book K.D. Tripathi. Here you can see at the first where one receptor has been shown, and this receptor here is a beta receptor. Just an example, because the agonist what they have taken, that is adrenaline or any adrenergic drug or any adrenergic neurotransmitter, right? So that will be act as an agonist for this particular receptor, and once the drug binds on the beta receptor, then what will happen, the G protein, in this case which G protein, GS type G protein. So usually when the CMP pathway is there, in this time usually GS get activated. So this GS when we say, it means G alpha. Okay, you remember that G protein has alpha, beta, gamma, where alpha is the active unit of it, and when the alpha is S type, means stimulating type, then that alpha will stimulate or activate the enzyme, effector enzyme. So that GS will get activated by converting GTP to GDP, so that it gets an activation, right? So now GS got activated, and this GS will activate the adenylyl cyclase. This is the effector enzyme here. Adenylyl cyclase will activate. Now this adenylyl cyclase, what this adenylyl cyclase will do, this adenylyl cyclase will generate the second messenger, right? Most of the cases the second messengers are going to be the kinase enzymes, means going to be activate the kinase enzymes, and then the kinase cascades begin. So it's all the game of the various proteins which are acting as enzymes in many places. So here you can see the ATP has been hydrolyzed by adenylyl cyclase and converted into AMP, because ATP converts into, you remove one phosphate from ATP, it becomes ADP, you remove one more phosphate from ADP, it will become AMP. And this AMP having a one chemical structure I have shown you in previous video, and when this chemical structure takes a cyclic orientation, it becomes a cyclic AMP. So this cyclic AMP is your second messenger. So now the cyclic AMP, which is your second messenger, is going to activate the further, or going to initiate the cascade of events from here, right. So what this CAMP will do, this CAMP will activate one kinase, right, and that kinase is going to be protein kinase enzyme, right. So this protein kinase enzyme is got activated here, usually in this case the protein kinase A, because these are various type of kinase enzymes are there. So here the protein kinase A enzyme is there, which is activated by CAMP. Now once this enzyme get activated, now this enzyme has various roles to play, right. In one way, this enzyme will go and activate some proteins and will enter into the nucleus, and there it will activate the Krebs protein, and that Krebs protein has a tendency to modify the gene, what's called the protein developments, right, protein synthesis process. So that way it happens one way. Another way, it can activate the troponin protein, it can activate the phospholipase protein, and the very important thing what it does, it is also stimulating the calcium, which is an intracellular stores of calcium, will release more calcium, right. And this, and also the activation of GS, as well as the activation of CAMP, as well as the activation of protein kinase A, they all have the role on calcium channel. They open the calcium channel and the calcium entry increases. Now this entry, which calcium which comes in, this entry stimulate more calcium to come from the intracellular stores. Like that, more calcium availability will be there inside. You can say that the activation of GS or the production of CAMP has increased the influx of calcium, right, intracellularly. So now calcium is more. This calcium will take part into the contraction mechanisms. Okay. So here what happens, it will bind with the troponin, and then the excitation contraction coupling will be there, and the increased cardiac contractility. If you are talking about the, if suppose that this cell which has been shown in front of you, suppose that this cell, this entire cell is cardiac cell, right. This entire cell is a cardiac cell. So what will happen in here, the cardiac contractility will increase, because the cardiac contraction mechanisms in the cardiac cells or the cardiac muscles will increase, right. Another way, it has various roles. So I'm talking about few. In the coming slides I'll tell you more roles of it. And if it is acting on the, if it is activating phospholipase protein, if it is activating phospholipase, then what will happen, the faster sequestration of calcium in sarcoplasmic reticulum. Here it will be the opposite effect. The calcium which came out from the sarcoplasmic reticulum, now by activating phospholipase, this calcium will go back into the sarcoplasmic reticulum, right. And then what will happen, a faster relaxation will be there. So in the one way, by through troponin, faster contraction will be there, through phospholipase, faster relaxation will be there. So this is what the protein kinase enzyme does. It will activate various proteins, and through various proteins various effects will be produced, right. So here if you see one more thing here, as you remember, the different alpha activation will do the different functions. If in the case of muscarinic receptor, suppose that here if it is a muscarinic receptor, then which alpha is going to be activated? Suppose that the drug binding on the muscarinic receptor, which is M2 receptor. So, which alpha is going to activate? GI, right. So, here GI is going to activate, and I means inhibitory. So, I will have the opposite effect what GS has produced. So, GS has activated the adenylyl cyclase to produce the CAMP, while GI will not do this, right. So, it means GI is inhibiting that CAMP formation, right. So, GI won't get activation and then it will not happen. So, like that the two different agonists binding on two different receptors, both are G protein coupled receptors, but having the two different effects here, right. So, the major effector here, major effector enzyme will be adenylyl cyclase in the case of this CAMP pathway. So, this way the whole CAMP pathway acts. If I tell you what are the functions of this CMP, or how, which are the proteins it is activating. So, you can understand here that the CMP dependent protein kinase A phosphorylates and alters the function of many enzymes, that I have already told you, ion channels, transporters and structural proteins. Why? To manifest as increased. Now, these are the actions what the pKa will do. Contractility, impulse generation. So, it will take care of the impulse generation on in heart, relaxation on smooth muscles, glycogenolysis process, lipolysis process, inhibition of secretion and mediator release, modulation of junctional transmission, hormone synthesis, and so on. So, like that the same GPCR by activating the pKa will do the n number of physiological actions in the body. In addition, CAMP directly opens a specific type of membrane calcium channel called cyclic nucleotide gated channel in the heart, brain and kidney. So, this is a one more additional information for you. Sometimes CAMP without using the pKa, right, not activating the second messenger, activating the protein kinase A, CAMP, which is your second messenger, directly opens the calcium channel by in the heart and brain, that is called as the cyclic nucleotide gated channel, CNG, right. Now, here is a very good example to understand the physiological actions of the CAMP and the CMP pathway. Just look at it here, you will understand very well. You see, in the case if there is an epinephrine or glycogen or any other agonist binding on the receptor, then what will happen after the receptor activation? You can see here, after the receptor activation, adenylyl cyclase, which is an inactive form, get into the active form, right. So, adenylyl cyclase got active. Once adenylyl cyclase become active, what it will do? It will activate CAMP, right. Now, CAMP has been activated. Now, what CAMP will do? You see that the third step, it will activate. It is an inactive protein kinase A, and after the CAMP it will become an active protein kinase A, right. Now, once the active protein kinase A formed, now this protein kinase A will act on certain enzymes to control certain physiological actions. So, here this protein kinase which has been activated here is going to activate one enzyme called phosphorylase, right, sorry, phosphatase. So, phosphatase enzyme is get activated. Now, you remember your biochemistry class of carbohydrate metabolism, there you will remember this. This phosphatase enzyme get activated. Now, it was inactive here, now it become activated. Once the phosphatase enzyme get activated, here what is happening? So, it will activate the glycogen synthase, right. In this particular part, it will activate the glycogen synthase. Here also, if you see, it is here, if you see below in this region, you can find that it is activating the another phosphatases here, or you can say you can activate the phosphorylase kinase enzyme. After activation of the phosphorylase kinase enzyme, here you can see the phosphorylase which was means which is getting active. Once the phosphorylase enzyme get active, it will convert the glycogen into glucose, right. So, like that, this one sided this process is happening by when the protein kinase A activates, it will activate the phosphorylase kinase enzyme, right. And in the other part, if you see this side, you can understand that when the protein kinase A activates, this protein kinase A will enter into the nucleus, here enter into the nucleus, and in the nucleus it will activate the Krebs protein, right. Krebs protein, this is a protein which is an intracellular protein, the Krebs protein get activated. Now, this Krebs protein will bind on the DNA, right, and is a DNA responsive element. It binds on the DNA and it will modify the mRNA. Now, the protein synthesis will be modified. Once this mRNA is being formed, which is a modified mRNA by the activation of Krebs, now this mRNA will go and do the protein synthesis. So, when it will do the protein synthesis, the different proteins will be formed, and that is what, when the proteins will be formed, that is what the effect of the agonist which has bind there. So, you have seen when the agonist either it is a epinephrine or the glycogen or any other thing which has bind on the receptor, it will do multiple actions, right. Here you can see it does this thing, here you can see it is acting on the glycogen and converting into glucose. So, like that, many physiological actions will be performed just by binding of one drug on the receptor, right. Then this all is the molecular mechanism which you can see here, this all is called as the molecular mechanism of that particular drug, right. So, unless until you do not know about this, it is very difficult to understand that how actually the drug acts. Okay. If I tell you that this particular agonist is increasing the blood glucose, suppose that I told you this particular drug is increasing the blood glucose level, then how it is happening? Now, you can explain this way, that it is binding on the receptor, activating it, and then activating the adenylyl cyclase, then adenylyl cyclase makes the CMP, then CMP activates the protein kinases, then protein kinases will activate the phosphorylase kinase, and then the glycogen will be converted into the glucose, and like that the glucose level will rise. So, this way you have to explain the mechanism of any drug. I hope you understood what I want to convey to you here. Now, to give you a little more clarity of the functions here, I have given you this diagram. Here you can see the agonist, in this part you see the agonist is binding and on the receptor, and the receptor has activated this G protein. Once the G protein get activated the adenylyl cyclase makes this CMP. This CMP will activate the protein kinase. When the protein kinase get activated, it will do these following actions. Which are those actions? Here you can see. It will activate the lipase enzyme. Here inactive lipase become active lipase. Once the lipase become active, you can see the lipolysis will happen, lipolysis, and you see in below when the glycogen synthase enzyme which was active become inactive. So, here is inactivating the glycogen synthase, then what is happening is the reducing the glycogen, reduce the glycogen synthesis, right. And if you see below, you can find that increased glycogen breakdown in the different case towards different enzymes, right. Glycogen synthase will activation, glycogen synthase deactivation will reduce the glycogen synthesis, and phosphorylase kinase activation will increase the glucose 1-phosphate enzyme, glucose 1-phosphate production. So, like that it will perform various actions just by binding on the GPCR, and then GPCR will follow the CMP mechanism to perform this particular action. I hope you understood this whole process, how it's happening. Now we will move to the second pathway. This is the phospholipase C IP3-DAG pathway. So here in this pathway we are not going to talk about the CMP, adenylyl cyclase, or the other proteins which we have discussed before. Here different things will be there. You can see one small diagram kind of I have placed here. This will help you out to understand that how this PIP2, which is present in the membrane, is going to play a very important role here, right? So what is the role it's going to perform? I will let you know. So here the PIP2, which is present in the membrane in the presence of phospholipase C enzyme, phospholipase C, right? This also various type of phospholipases are there. If you remember your lipid metabolism chapter in your biochemistry, you will get this idea. So phospholipase C is going to hydrolyze this PIP2, right? Once this PIP2 will get broken, it will form two things, one inositol triphosphate and diacyl glycerol, right? Now these are two very important components which are going to deal with this pathway. Let's see. Here you can see GPCR is there. You can find the GPCR, and this GPCR is get activated by a drug. So your drug, which is going to be your first messenger, is going to bind with the GPCR, and after the activation of GPCR, this here you can see this got activated. It is activating phospholipase C enzyme. In the previous pathway you have seen, in this place adenylyl cyclase got activated, right? But here the things are different. Here it is going to be phospholipase C which is going to play the role here, phospholipase C. There are phospholipase B are also there, right? So but in this case phospholipase C get activated. Even in some cases phospholipase B has the conversion into phospholipase C as well, right? So now phospholipase C got activated, and this activation has broken down this PIP2, which is already present there, in the membrane. See, one more thing I want to make you understand. This whole process is happening in the membrane and just below the membrane. So everything, that's why these receptors called as the membrane bound receptors, right? Because they are performing their actions within the membrane or below the membrane, right? So the G protein coupled receptor which is present as a transmembrane, but the other thing, every whole process is going just below the membrane, right? So here once the PIP2 get activated, this PIP2 will form two components, DAG and IP3, right? So DAG is a diacyl glycerol and IP3 is inositol triphosphate, and both of these DAG and IP3 will act as a second messenger here, right? And now they will perform various actions by the same method. So what is happening here, this IP3 will activate the calcium channel, right? Once it will activate the calcium channel, the outside calcium will start coming in, right? So more calcium will be there inside, and here the calcium is also playing a role as a second messenger, because once the calcium is been activated, this calcium will activate more calcium, or you can say this calcium will attract more calcium to come in, right? Which is maybe from the internal stores, and once this calcium releasing, this calcium will do various functions by binding various proteins, right? By activating various proteins here. So this way it is performing. Here you can see second messengers combined with the calmodulin to activate myosin light chain kinase. Here another protein will come, that is the MLCK, myosin light chain kinase, and that myosin light chain kinase, you remember the myosin, actin and myosin in the muscle contraction mechanism, right. So, here myosin light chain kinase enzyme is going to get activated and binding with the calmodulin protein, and then it performs the contraction mechanism, right. Another important regulation of calcium calmodulin protein kinase C, protein kinase, that is called CCPK, that is also going to play a role, that I will tell you in the next slides. So, I hope you understood how this phospholipase C is playing a role here, right. Here also the diagram will give you a little more clarity. You can find here that once the phospholipase C get activates, it will hydrolyze this PIP2 into IP3 and DAG. Now, this IP3 will go and act on this calcium channel, right, calcium gated channel, and it will bring more calcium in, and here the DAG is going to act on protein kinases, right, this protein kinase C. Now, this protein kinase C is also get activated, that is why the calcium is also called as the second messenger, because once the IP3 activates the calcium channel, then the calcium, because it is an IP3 gated calcium channel, means when IP3 binds on the calcium channel, the calcium channel will open, and when the calcium channel will open, the more calcium will come in. When the more calcium will come in, the calcium itself, here you can see the calcium itself is activating the protein kinase C. So, that is why here in this case we call the calcium also as a second messenger, right. Hence, another way the PKC is also activating the protein kinase C here, right. So, here you can understand that when the DAG has activated the PKC or the calcium has activated the PKC, now this PKC can follow various pathways to perform its action, right. PKC can activate the RAS protein, right, and the RAS protein will activate the RAF protein, RAF protein. RAF protein will activate the MEK protein, MEK, right. You can understand that once the PKC activates, it may activate RAS protein, you can say RAS protein. This RAS protein can activate the RAF protein, usually RAF1, and this RAF will activate the MEK protein. This MEK will activate the ERK protein, right. And like that, the ERK will further go and activate mTOR protein, mTOR, right, mTOR protein, and likewise this mTOR protein finally changes into the DNA synthesis, will change something in the DNA. Likewise, it can change the protein synthesis, finally protein synthesis, right. So, likewise the one pathway follows. Sometime the PKC will not activate RAS, it will directly activate the RAF. Sometime it will directly activate the RAF, right. Here it will directly activate the RAF, and through RAF it will follow the same pathway again. Okay. So, understand this point, once the protein kinase C get activated, it had various routes to follow, and then it will do the required action. Sometime it has another pathway to follow. Once the PKC get activates, this PKC will activate the PKD, and through PKD it will activate the COX protein, right, COX2 protein. This COX2 protein is also rolled into the PGE2, prostaglandin E2, right. And sometimes this PKD also has a role to activate NF K beta. Okay, so various ways are there. By the various methods they are following, they are controlling various physiological mechanisms, right. So, you should understand this point very clear here, that once the PKC get activates, it will activate the multiple processes, right, and the multiple physiological effects it will produce, right. In the next slide, I will tell you how this is the diagram which tells you how this phosphatidylenoacetol bisphosphate is going to convert into the phospholipase A2 will convert it into the DAG and IP3. So this is the here this you can see PLA2 phospholipase A2 will act on this and will produce the DAG, right, and the other part of it will be acting as IP3. So like that this is the structure of the IP3, this part, right, and this part is going to be the DAG. So like that the PIP2 will be get broken or hydrolyzed by the phospholipases and converts into the IP3 and DAG. Okay. So I hope you understood this pathway, what I want to make you clear. And usually in the case of, as I told you, when the GS or GI get activated, they usually follow the adenylyl cyclase CMP pathway, and when GQ or G11 get activated, they usually follow the phospholipase C pathway. So like that different activation of the different alpha protein depends which second messengers or which effector enzymes going to get activated. Okay, so now I will move on to the third pathway, which is the channel regulation. So you know very well that channel regulation do not need any second messenger to get involved with. So here without second messenger this process is going to happen. Let us see. So here I tell you few things before showing you the diagram. The activated G protein can also open or close ionic channels. That is it mean G protein not only activating the effector enzyme and then the second messengers and third messengers, while it can have the role for opening of the calcium channel, potassium channel, sodium channels, like that, right, without the role of, means directly the alpha will activate it, right. For example, like CMP or IP3, and bring about hyperpolarization, depolarization and changes in intracellular calcium. So likewise by only controlling the calcium, by only controlling the opening or closing of the channels, the physiological roles will be controlled. For example, the GS opens calcium channel in myocardium and skeletal muscles, while GI and GO opens potassium channel in heart and smooth muscles, as well as close neuronal calcium channels. Okay. This diagram will make you understand here what happens. This is the receptor ion channel, this is ion channel, and this is a receptor, right. So only two things are there, but there are in the coming slides I will discuss about the receptors which are directly acting on the channels. They do not need the GPCR, but here because it is a GPCR involvement is there, first the drug suppose that this is a drug or a signaling molecule or a component or a chemical or neurotransmitter, anything is, or agonist which we call it as, or ligand which we call it as, is going to bind with the receptor here. This is a green color of your GPCR receptor. This alpha, beta, gamma, these are the G proteins, right. So once the signaling molecule binds on the receptor get activated and the G alpha is get activated here, and this G alpha is activating the any membrane bound effector, right, and it can be any component which is going to activate the ion channel, which is going to open the ion channel here, right. So sometime it needs any membrane bound effector to activate, or sometime directly it will G alpha will activate the receptor ion channel. So usual cases we believe that this activated G alpha is directly going to activate the ion channels, right. Once it opens the ion channel, the ions which are present on the upper part, they will start coming in, right. And once these ions come in, now further role will be played by these ions only. When these ions come in, the ions will either do the polarization, hyperpolarization, depolarization, or various processes they will perform, and the physiological effect will be produced, right. So, this way it is happening. Here I want to tell you one thing. These receptors, these are called as the receptors without having the second messenger intervention, right. So, but sometimes they activate the effector, any other effector, and through effector they activate the ion channel, and sometimes directly they activate the ion channel. Now, this is the way this third mechanism of the G protein coupled receptor has been performed. Now, this is a summary sheet, you can find it in the book A.D. Tripathi. Here it shows adenylyl cyclase CAMP. See, all these three pathways have been shown here: adenylyl cyclase CAMP pathway, phospholipase IP3-DAG pathway, and channel regulation pathway, right. So, here it has been shown like these drugs. If you see adenergic beta receptor, histamine H2 receptor, dopamine D1 receptor, glucagon, FSH and LH, ACTH, TSH, prostaglandin, EP2, prostacyclin, and adenosine A2. So, these all drugs and their receptors, what they are doing? They are activating the adenylyl cyclase. Okay, it means they are acting through GS. They are acting through activating GS. While at here, if you see here, adenergic alpha 2 receptor, muscarinic M2 receptor, dopamine D2 receptor, and 5-HT1, GABA, AB, opioid, and so on are suppressing the adenylyl cyclase. So, once they suppress the adenylyl cyclase, usually they are acting through GI, right. So, they are suppressing the adenylyl cyclase. Okay. Here if you see the next one, the phospholipase IP3-DAG. So, here also it is shown that these drugs are acting through IP3-DAG pathway. Third one is the channel regulation. Here you can see the adenergic beta 1 receptor is actually acting through the calcium channel. Here dopamine, GABA, opioid, they are acting suppressing the calcium channel. They are decreasing the calcium channel. Here they are activating the calcium channel. And there are other drugs like dopamine D2, 5-HT1A, adenergic alpha 2, they all are activating the potassium channel and playing the role, right. So, whenever activation of calcium channel is there, the depolarization will come. When the activation of potassium channel will be there, then hyperpolarization will be there, right. So, this way these all the drugs are specifically using one or the other pathway to produce their physiological responses. Here also the various subtypes have been shown, right. So, if you have the G alpha S, or you can call it a GS, or GI, or GQ, G11, GQ 12/13, G beta and gamma. So, you can find out what their roles, what are the effectors they are activating, and what are the different isoforms they are having. These isoforms have been found by the various gene cloning mechanisms. So, you can see if G alpha is there, G alpha S, or you can say GS get activates, then adenylyl cyclase and SRC family tyrosine kinases get activated. When GI get activates, then adenylyl cyclase and adenylyl cyclase decrease and phosphodiesterase increase. Like that, the different type of alpha activation or deactivation will change the activation and deactivation of effectors, and so on their physiological role. Here you can find GS, GI, GO, GQ, G13 has been shown what they are doing. They are doing adenylyl cyclase activation, calcium channel activation, and likewise what I have already told you. Here you can find the next one. It shows the various receptors and their is coupling where they are activating. So, if it is a muscarinic receptors, they act on GI, GO and GQ, right, different effects on the different places on the different organ system. Dopamine acts on GI and GO. Likewise, different drugs are having their affinities towards the you can say not affinities, you can say they bind on all bind on the same G protein, but the activation of the alpha is different, right. So, likewise, if the activation of alpha is different, same way the responses what these alpha and the physiological role what these alphas are producing are different. Okay, so I hope you understood everything what I told you, that from the previous video there was a structure of GPCR, and in this video you have seen the 3 different pathways what the GPCR is following. So, number 1 was CAMP pathway, second one was IP3-DAG pathway, and third is the channel regulation. So, likewise, these are the 3 main mechanism of GPCR activation. Now, we will move on to the another receptor, means first one was the GPCR, G protein coupled receptors, right, and their mechanism of action. Now, the second receptor, moving on to the second receptor, that is receptors with intrinsic ion channels. It means what? There is a receptor and the receptor itself is having one ion channel in it, right. It is not like GPCR, that first GPCR will get activated and then the alpha will go in some other place and then activate the channel, but here the receptor itself is having a channel, right. So, like one part of the receptor is a receptor and another part of the receptor is a channel, right. So, likewise it is an inbuilt channel in it, right. So, let us see how this is happening. So, I will explain you about this receptor how it look like. It's a structure, and then I will tell you how these receptors are acting and their functions. So, these are also the transmembrane receptors. They are also called as ionotropic receptors or called as the receptor operated channels. They are also called as receptor with the intrinsic ion channel, transmembrane ion channel receptors, various ways you can call them. And these receptors can act excitatory or also inhibitory, right. If the cations are binding on them, they are generally excitatory. When the anions are binding on them, they are generally inhibitory. So, like that they play their role. Now, these are cell surface receptors, right. So, when these are the cell surface receptors, they are present on the surface, which are ligand gated ion channels. So, these are called as the ligand gated ion channels, right. So, these channels encloses these receptors encloses ion selective channels for sodium, potassium, calcium or chloride within their molecule. So, that is what I told you, the receptor itself is having a channel, right, and these channels can be for sodium, potassium, calcium, chloride, right. So, if you see the structure, you can find that this is a 5 rosette like structure, right. You can see this part, this is the upper view. From the upper side, if you see, it will look like this 5 rosette structure, right, 1 2 3 4 5. So, 5 rosette like structure. These are also the transmembrane proteins are there which are making this structure, and these structures this structure has been divided into like we call it as an alpha, beta, delta, one more alpha and gamma, right. So, you can say 2 alpha, 1 beta, 1 gamma and 1 delta. Likewise, these 5 rosette like structure will be formed. And another very important thing you can understand here, that the agonist is binding only on 2 places, right. Agonist binding site is available only on 2 places. One, gamma and alpha junction, and another sorry, alpha and gamma junction and alpha and delta junction, only two places it is give it means the agonist can bind. So, agonist binding site is available only on these two places. In the structure, in the left side, if you see in the left side, you can find the from the front, if you see, it will look like this. So, alpha, beta, gamma, deltas are arranged in such a way, and in between there is a 0.7 nanometer, this is a 0.7 nanometer pore is available. So, when this pore is open, we call it as channel opening. When this pore is closed, we call it as channel closed, right. So, that once this the pore opens, the agonist or the drug or the ions will move inside. Not drug is binding on this particular place, right. Once drug is binding and opening the channel, then the ions which are available outside will use this pore to come into the cell, right. So, they will come intracellularly. Here you can understand very well. Suppose that sodium is, suppose that this is an example of a sodium channel, right. So, this sodium will use this pore and come inside, right. Once the acetylcholine as a drug, as agonist binding on this sites, binding sites. Okay. So, likewise this all arrangement is there, and once the, so you can see this is a receptor which is having a ion channel inside, right. So, where these are the drugs are binding, this all is the receptor part, and inside it is having a function like a channel. So, once the agonist binds on the binding site, agonist binding site, the receptor will get open, this 0.7 nanometers diameter, the pore will open and the sodium will come in. In various cases, it may be the potassium channel, it may be the chloride channel, it may be the calcium channel, right. But here the example shown for the sodium channel. Understood? So, now if you see the examples of the various examples of the same receptors, you can find like there are the inhibitory category of receptors, for example, GABA A, glycine, they all are the inhibitory inhibitory receptor of the same. Another excitatory receptors are also there, glutamate, in this kainate, NMDA, AMPA type receptors, 5-HT3 receptors, cholinergic and nicotinic receptors, they all are the excitatory type of receptor with intrinsic ion channel, right. The response are faster with these receptors. One thing is very important to note, that these are the fastest receptors, right. They act in milliseconds, right. So, likewise, and these channels are involved in rapid synaptic signaling. If you see that in brain, a lot of these receptors are present, because brain needs a lot of faster actions. So, in the synaptic signaling signals messaging between the synapses, these kind of receptors are available, right. So, in the rapid synaptic signaling between nerve cells and other electrically suitable or excitable target cells, for example, nerves, muscles, these receptors are present, right. These channels are all built from homologous polypeptide subunits, which assembles as pentamer. So, if you see the structure, it looks like a pentamer structure, it's a pentamer structure, right. Glutamate gated ion channels are an exception, right, in that they are constructed from a distinct family of subunits and form tetramers resembling the potassium channels. So, little difference has been observed in the case of glutamate gated receptors. They are also the same receptors, but in place of pentamer they are making a tetramer. Okay, that is a little difference. For each class of transmitter gated ion channels, there are alternative forms of each type of subunits, which may be encoded by distinct gene or else generated by alternative RNA splicing of a single gene products. It means, as you have seen there, the alpha has a various forms, G alpha, sorry, alpha, G, GI, GS, GO, GQ. Same way here also the subunits are of different types, which are making the various type of the same receptors. So I hope you are clear about the this type of receptor, which we call it as intrinsic ion channel. This is another diagram, this diagram from KD Tripathi. You can find this alpha, beta, gamma, delta units clearly here, and you can see this is a transmembrane receptor. It starts from the outer side and goes up to the inner side. This is the currently the first diagram shows you the closed structure, and the second diagram shows the open structure, where the upper rosette like part is little wider and the pore is open, and the sodium, potassium and calcium and other ions can enter into this. So I hope you understood these this receptor, and the next two receptors I will cover in the next video. That will be easy because it is become little bigger video, so that's why I opted to give it into the next video. The other two receptors are very important receptors, and here you please go through conveniently about the GPCR mechanisms plus intrinsic ion channel and its structure and mechanism of action, how it works. So these are the references I have used for making this content, and thank you very much for listening me so honestly. Thank you.